Hi, this is Jason Delborn. Thanks for some great presentations. I wonder if you could address the question of whether, if gene flow is considered as part of EAs or other processes of risk assessment, how gene drives change the calculations of oversight of gene flow, given that a gene drive, if a few organisms were to escape, that there'd be the potential for that gene to reproduce quite broadly. So I just wonder if you could address that issue of overseeing gene flow and how gene drives might change that oversight mechanism.
Should I can go first. I think that's something that's definitely going to affect us. I think we're still trying to think through that one. What you saw in my presentation was that everything we do when we're regulating organisms is aimed at containment and confinement. And gene drives are specifically intended not to be confined once they're released into the environment. So this is going to be very new territory for us that we are just really beginning to explore. And unfortunately, I can't really say much more than that because it is so new for us. But it is going to, I think, require us to think about things in a new way, perhaps more analogous to releases of biocontrol organisms. Perhaps I heard some suggestions this week at the Society for Risk Analysis about thinking about invasive species as possible models. So these are all things that we just need to think about still.
This is Larissa. I think I'd like to echo a lot of what Alan said. And for us in particular, because we do things on a case-by-case basis, it would depend a lot on what we anticipated the hazard and the risk to be. Genes flow two ways. One is through sexual reproduction, and the other is through asexual transfer. And what we're talking about with respect to gene drives is sexual reproduction, but biased reproduction, so that you're talking about a kinetic difference as opposed to, if you will, the thermodynamic question of whether it will or it won't happen. So again, it would be dependent on what the intended use of the gene drive mechanism is. If it's intended to be freely released, then clearly at the end of the day, you don't have so many concerns about it getting out into the environment because it's supposed to be in the environment. If it's supposed to be a contained use, then you have to look very carefully at whether or not you have the appropriate physical and biological containments in place.
The only other thing I would add is in terms of our deregulation procedure, even there, I mean, obviously we do consider gene flow to sexually compatible relatives there. For the most part, most of the crops that we've deregulated so far, many of them don't have, like corn and soybean, which is the overwhelming majority of what we've deregulated, just don't have sexually compatible relatives in the U.S., so it becomes a non-issue. Some of the others do, and there we might, we'll consider potential impacts should a gene get into a sexually compatible relative, and we assess things like the likelihood of that gene flow and so on. So in that sense, we do have some experience with it, but again, it's not in a gene drive system where you're actually really intending to spread this through a population.
Thank you. Other questions? One of the things that has been mentioned across a variety of the sessions that we've held is the simplicity of using CRISPR technology and how high school students and others, maybe not home biology kids, but a more non-institutionalized kind of science may be possible with CRISPR and potentially leading to a more independent kind of gene drive research. Can you all speak to the broader question of the fact that much of the regulatory structure that we have is aimed at institutional oversight as well as, more so than individual researchers who may not be subject to institutional oversight? Is there a way to think about the more independent researcher who may be working in a garage, for example?
Yeah, so with FDA, it doesn't matter whether you're in an institution or in a garage. If you have an article that has not been approved, then you cannot enter it into commerce. And we have a fairly broad interpretation of the Commerce Clause. I think the whole thing about the DIY bio and biohackers and BioBricks and the iGEMS competitions and all those things should be stimulating us to think about surveillance and compliance in a different kind of way than we have in the past. But I think, the statutes are the statutes, the regulations are the regulations, and again, it doesn't matter if you're working for industry or in an academic lab or your garage or a rented space that you do experiments in. You're still subject to the same strictures that are applied to anybody else.
Yeah, I would add that for our 2014 report, one of the main challenges that we identified was that there would be many more microbial products, in large part based on this democratization of biology and the DIY bio people and just coming out of other types of labs. And we identified that as a challenge for EPA just in the manpower and breadth of expertise that they were going to have on board to deal with all of the applications that were coming. It seems to me there could be an analogous thing here with the animal applications. I mean, Larissa has described the regulations really well, but there have been very few animal applications that have gone through that process. And it's been very case by case, so much so that it's hard to predict exactly what the next process will look like. And so I think there could be just a challenge for the agency, and I mean, clearly the applicants should go through that process, but the agency will have to deal with multiple different applications, different types of organisms, different animals. I think it would be up to the agency to be on top of it and be able to process those things at a reasonable pace.
Sarah, I'm not entirely certain what you mean by that. And we've got multiple applications and multiple different types of species, and everything's done on a case-by-case basis. I think that I don't think any one approval presages how we're going to deal with a completely different organism and a completely different construct and a completely different use. And because technologies are new, it takes time to develop a track record. But I mean, there are lots of challenges that are presenting themselves because these are emerging technologies. But I think to a first approximation, I don't envision that we don't have the regulatory authority to deal with just about all of them.
Larissa, I agree with you. I think that you do have the regulatory authority, and the assessment like capture that comes. The problem is that, do you have the manpower, the funding, to handle 20 different applications that may come up in 1 year? And I mean, I don't know, maybe you all can scale up as needed to deal with those things in a reasonable time frame. And I hope that you can.
I would just add from Avis's perspective to that, the big challenge here, I think, is building awareness in that community of what regulations actually might apply to what they're doing, and us as a regulatory agency building our awareness of what's going on in those communities too. And I think that's a big challenge for us. But that said, I mean, at the moment we are beginning to get actual applications from people in DIY communities and so on. That's been dependent on our ability to make contact by hook or by crook with people who we hear about, or people who happen to hear about us. So I think perhaps what we need is a little bit more deliberate effort at reaching out to those communities and engaging in that dialogue. I know the security community is doing that very well.
We are almost at the end of our time, but you just said something I'd like a little bit more follow-up on, if you would. You're beginning to get applications from people from the DIY bio community. Well, they're not a community; they're individuals, obviously.
So,
but what does that look like?
So, I mean, the example I'm thinking of in particular, and the committee members may, or all of you may be aware of it, is the Glowing Plants project that got Kickstarter funding a couple of years ago to engineer fluorescent Arabidopsis plants, and part of their Kickstarter funding effort was if you contribute a certain level of funding, you'll get a kit where you can make your own. And so we worked with that. The glowing plants that organization was making themselves, they used bioistic transformation methods, so actually did not trigger our regulations, but the kit uses agrobacterium, so it does trigger our regulations. And so that organization has actually been trying to assist people who are trying to get these kits to submit applications to us for movement permits, essentially. And we've received about a half dozen of those so far, and we're working through them. I will say it presents us with new situations. We're used to dealing with universities and companies and institutions with containment facilities and so on, and you know, some of the people who are requesting permits may not have that. So we're kind of working through that process.
Thank you. That was very helpful.